Alright, this presentation is about antibodies and the ELISA test. Antibodies are proteins created by the body during an immune response to a specific antigen, and the ELISA test is a method used to detect the presence of an antigen in solution. So, in the body, when immune cells called B cells are introduced to antigens, they can bind the antigens and then they will ingest them by phagocytosis and digest them into smaller pieces, which they can then associate with these MHC molecules that they display on their cell membrane. Um, Once they're on the membrane, they can be recognized by a helper T cell, which can then bind to it and secrete interleukin 2 or other cofactors that pretty much activate the B cells, and then the B cells will then divide into memory B cells, which will be used for a later response. But then effector cells called plasma cells, and the plasma cells make antibodies, which then go to the bloodstream and fight the infection. So antibodies have this kind of Y-shaped structure where there is this long, heavy chain and the shorter light chain. This area of the long chain is constant, and then this area of the short chain is also constant. So the variable region is this area in blue, and this is where the antibody binds to the antigen. Um, so the ELISA test makes use of antibodies in determining the presence of antigens within body fluids. So you have antigens, a primary antibody, a secondary antibody, which then binds to the primary antibody, and the secondary antibody will usually be labeled with some sort of enzyme, and then you have a substrate. So here we have the primary antibody binding to the antigen, and then here we have the secondary antibody binding to the primary antigen. So when you are introduced to an antigen, you have this immune response, and the B cell makes antibodies. We can take those antibodies and then inject them into an animal such as a rabbit, and the rabbit will, or the animal, will have its own immune response to these human antibodies, and it will create its own antibodies right here. So these would be the secondary antibodies. So then we take the secondary antibodies and then we can bind them to an enzyme, just a covalent bond, where now this is labeled with this enzyme. The enzyme will, when introduced to a substrate, cause a color change. So here, this is, we have a body fluid with antigens within it. We put these into these wells. It's like a plastic strip of wells. Here's one of them zoomed in. Oops. So here's the fluid with the antibodies. So when they're in here, the antibodies will bind to the plastic wall of the well. So then we will rinse it out and get everything else out of there and be left with just the antibodies bound to the well. It's just hydrostatic interactions that cause them to bind to the wall, so they stay in there. Then we add the primary antibody, which will then bind to the antigens. We rinse it again, and then we add the secondary antibody that is bound to this enzyme, and then these will bind to the primary antibodies. And we rinse it again, and then in the final step we add the substrate. When we add the substrate, it will react with the enzymes that are bound to the secondary antibodies and cause a color change in the solution. If you see a color change, then that means that you know that the antigens were present. If there's no color change, then the antibodies or antigens were never present. As shown here, there's no antigens here, so you get no color change. So here are the results of an experiment done where we can see that these first 6 were the control group. So we knew that these 3 were positive, and we knew that these 3 were negative. So you would expect a color change for the positive ones and not for here. So here, these were the tests, and these wells labeled 5 did not change color, and the wells labeled 6 did change color. So by comparing them to the control, we can see that the wells labeled 5 did not contain an antigen, and these wells did. So the ELISA test has a lot of uses. It can be used to detect diseases in animals and plants. And it could also be done to detect drugs and to determine pregnancy. So it has a lot of beneficial uses, and it's used a lot.